And also thank you, Marcus and Hilda, for giving me this slot in the very last minute. So I am not a synchronization person, I'm not a neuroscience person, and I'm not, well, I know dynamical systems, but I wouldn't say I'm a dynamical systems person. So what I thought I could do, especially given that this is before lunch on Sunday and the last part between you and a nice meal, is to give you a little bit of some ideas on work we are doing on collective behavior, but from a different approach. So hopefully maybe we can discuss if some of you is a collective behavior person or an active matter person, because I think there are many, many opportunities to work on these systems that I'm going to show you today. So as I told you, since I am not let's say very good fit for the school or for the workshop, I wanted to give you a bit of some examples of questions I'm interested in answering, in case some of you is interested as well. I will be around for the whole next week too, so maybe we can chat more, even I don't know, even if you have not been thinking about the systems but just want to learn more. So I am very interested in general in studying emergence in ecological systems, in very different ecological systems. For example, in bacterial biofilms, we study how different strains arrange when bacteria grow in colony, and how, depending on the traits of the bacteria, we get different spatial structures, and what are the implications that this has for things like virulence of these strains and infection, evolution of infection on time. Very recently, we've been working a lot on trying to understand what are the consequences of movement for populations of animals. So we have a very large data set of animals that we and some of our collaborators have been tracking for many, many years, different species in very cool places, and basically we do stochastic process modeling to how these animals move, trying to develop methods to estimate how they occupy space. And more recently, we've started to work on how these spatial patterns of space occupation lead to different population dynamic processes. We also do some work on vegetation dynamics. So how is that plants, when they have little water, form these very regular patterns that are kind of Turing patterns. And recently, we've been also working a little bit on algal blooms and how is that fluid dynamics interacts with ecological dynamics to lead this burst of plankton in different regions in the world. So if you want to talk about any of this, just let me know and we can talk, not necessarily today, as I said, can be tomorrow if you are very tired today. Great, so today I told you I was going to talk a little bit about collective behavior. That was the promise I made to myself. And since I was so thankful to Hilda for this opportunity, I wanted to start with a slide that I know she will like a lot, giving you two examples of collective behavior systems that probably have opened many collective behavior talks. So you have on your left side an example of a fish school where fishes, because of the way they interact with each other, they form these torus. This is very similar to what Carmen was explaining you last week, but this is recorded in the wild. This is in the ocean. And on the other side, you have like an example of a flock of starlings. I think this one is from the sky in Rome, but you've probably seen many examples of these pictures, both in lectures and in papers. So people have modeled this problem from different approaches. We've seen a little bit of self-propelled particles with different interactions to understand how these patterns emerge. That's a very physicist approach. Biologists built on that a little bit to build more complicated models, let's say, that at the end are fully computational. They try to introduce vision fields, vision angles, measure information transfer networks with very sophisticated experimental setups, and try to get models that are harder to treat mathematically. It's very large IBM simulations, and they aim to explore like how these fine-tuned details, let's say, lead to all these emergent phenomena patterns. So if you're interested into this kind of modeling approach, this very nice 2002 paper by Ian Cousin, I think it's a very nice reference. And built on that, you can keep reading on works, similar works, and you will see a little bit how interesting it is to establish connections between the active matter community and the interacting particle systems that for us physicists maybe are more familiar, and how biologists tackle this problem from a different angle. So just to move away a little bit on fishes and birds, I wanted to introduce you two systems that exhibit collective behavior that we've been working on, and the kind of questions that we were interested in addressing that are a little bit different to the maybe more common questions that one can ask regarding the emergence of spatial patterns. So the first system I want to introduce you is a very, very tiny system. These are microbes. This means that what you see there is a movie that was recorded in Thomas Gregor Lab, now I think 16 years ago or 15 years ago, more or less. So what you see here is a Petri dish. So it's a very, very small plate that you put under a microscope, and then you record the microscope. The resolution of the image is the one that we had 15 years ago, but I'm going to show you more recent stuff later. So I'm not going to tell you anything. I'm going to give you details afterwards. I just want you to see what's going to happen now in a while. You see everything is noisy, blurry, and now we are going to see some spiral waves that are start to form and travel around. You see that now they are going to become more stable. Yeah. And now eventually something is going to happen that is even more interesting, in my opinion, than this emergence of spiral waves. And you see that spiral waves collide in space. They create some kind of frontiers when they collide with each other, and that is going to define some territories, let's say. And then we have this streaming of white fingers, let's call it white fingers so far, just to not spoil anything. And these streams will converge to a very, very bright point in space. Yeah, so here the conserved number of cells is fixed. So these are amoeba. I'm going to now give you details on the biology, on what is happening here. I just wanted to show you how it doesn't even have a scale, and it's on purpose that I didn't put a spatial scale. I'm going to give you all these details now, and if I forget any of them, just please ask. Do they crack? No, nothing related to that. Let me give you the details of what is happening here. So as I told you, these are microbes. And they have a very, very interesting life cycle. So they are unicellular. When you saw the noise background, it was just cells that were uniformly spaced in this Petri dish. The point is that these cells were hungry, as you probably are now. So they were taken to starvation and washed. And what happens in this organism is that when these cells get hungry, they start signaling with each other. They start communicating with each other, and it is these chemicals what creates the traveling waves that you saw. And there is a lot of work on excitable media trying to study and to understand how these traveling waves form, and there is beautiful papers on that. Some of them even doing synchronization analysis, and I can share those with you if you want. And what happens is that once this signaling process occurs, then they form these bright points, and these bright points were aggregates of many, 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 many cells. And once those aggregates form, they start developing into this kind of pretty mushroom that you see here. That is probably my most artistic work ever. The interesting thing is that within this fruiting body, the cells that are on the top, they are going to be dispersed to a new place where hopefully they will find food. But the cells that are here, they are going to die. So this multicellular fruiting body has division of labor somehow. So then people found this a very, very interesting system to understand how is that multicellular life can evolve. Why? Because these cells, some of them are dying just for the others, and you should, pardon me, in using like this kind of human-like wording, especially if there is any biologist in the room. They are kind of sacrificing so others go to somewhere else. And this problem is especially important because sometimes cells within these different fruiting bodies are completely unrelated genetically. So you can mix two different strains, they will still talk to each other, and they will still do this work. So there's been a lot of work on how is that this aggregation process happen and how is that it evolved in time, basically. Evolve, I mean evolutionary timescales, not dynamics. But now let me show you more recent data with more quality and now with its proper spatial scale. And this is data that we collected for this project. And I'm going to show you what happens with a little bit more of resolution and zoom. Now you see that we have these tiny small drops. Those are, I wouldn't say single cells, but let's say, to the best of our knowledge, almost single cells. Again, these guys have hunger again. They are hungry. So this is what happens. They start moving around, trying to find food a little bit. Now we are going to see some intermediate clamping process that we still don't understand very well. But now you see this formation of the streams at a higher resolution and the formation of the fruiting of the aggregates. And then, yeah, I forgot to tell you that they form also this cool slag that starts moving on the soil, trying to find light. It's a very, very complex life cycle. What these amoeba do is crazy. And it can take you crazy as well if you try to understand it. So this is what happens at higher resolution. Now we see that everything that we were discussing before already happened. There is no cells here. Everything, we have the fruiting bodies, job is done. But what you see in the screen is that there are still dots there, right? There's still points there. There's some cells that are clearly still in the environment. I can show you an even higher resolution video where you will see that now it's clearly this, we have the single amoeba. And this is a zoom where you will see now the stream. You see to this part of the plate. And now we have cells there. And these cells are alive. They are still moving. Clearly, they will die fast because they have hunger and they were not able to disperse somewhere else. But they are alive for a little bit of time. So the question that we had when we started working on this project was what is going on with these cells that are not part of the collective behavior. Probably the first thing that comes to your mind, and what many, many of you are already thinking is that, come on, I mean, you have here like, I don't know how many thousands of hundreds of cells. It's noise. You cannot synchronize perfectly your population, and that's it. They are there, it's noise, they die, bad luck. That's one possibility, but what if they are not? What if they are something that is biologically meaningful for this system? So to understand if it's something that can be potentially meaningful for the system, the first thing that you have to do is to count these cells, right? And this is very, very interesting because when we started working on this project that has a strong experimental and theoretical part, there was no experimentalist in the team. So I started my postdoc, and they told me that there was this system. We started discussing about it, and I started doing some models, getting crazy. All the models were wrong, 2 years of modeling that was not very accurate, let's say. And then we thought we had to do experiments. And the experimental PI that was there told me, like, come on, I mean, all you have to do is to count cells. I mean, don't you know how to count? And he literally told me those words, like, you're a physicist, don't you know how to count? And the point is that I know how to count, but this project didn't need to count cells. It needed to learn how to count these cells. And this is an extremely challenging problem because amoeba are not regular, so it's not that you're going to count points. And as you saw, they are all over the place. Sometimes they stick to each other, and you have 4 cells. How are you going to treat that? So we were very lucky that I didn't do the experiments, both me and everyone else in the project. So we had a very, very good experimentalist that joined a little bit after, and he was able to count these cells. So what I'm going to show you now is on the y-axis, you have the number of loner cell densities. I'm going to call them loners because they don't join the group. And on the x-axis, what you have is the density of plated cells that we have in this experimental setup. So what we are going to do is, this movie that I show you, I'm going to do it many, many times, just changing the density of cells that I put every time in my experiment. And I'm going to count the number of cells that are left behind. This is going to show you a very, very interesting, give us a lot of very interesting information about the process that is leading this, the mechanism that is leading this aggregation process, because maybe some of you are now thinking, well, I mean, maybe these cells, they throw a coin, and with a probability P, they go to one side, and with probability 1 minus P, they stay. This is the easiest decision-making model we could come up with. If that was true, then this plot should show us a straight line with slope 1, with a slope P. or if the probability of aggregating is P, then 1 minus P, right? It should be a linear correlation. So when the density of cells is low, aggregation doesn't trigger. So there is no enough density for cells to start aggregating, and therefore all cells in the plate remain as loners. And that's what this 1-to-1 line means, this slope 1 here. Please take into account the different scales in the y and x axis. This is not a one-to-one proportion. However, what is really interesting is that when the density of plated cells goes beyond one threshold, what we find is that the density of cells that remains behind has no correlation with the density of cells that we had initially on the plate. What this is telling us is that the decision-making process behind this partitioning, population partitioning behavior is way, way more complicated than just flipping a coin. But we still haven't characterized what is it. We can repeat this experiment with different strains, and you see that now the data is not so beautiful, there is more variability, but still if we do a statistical analysis, we see that there is no correlation, and we can somehow reconstruct a probability distribution function for this density of loner cells that is conserved across replicates of the experiment. So this is something that is very important. Biological systems are inherently variable, have inherent variability. So that means that you have dispersion, or the fact that you have dispersion around this straight line doesn't mean that you don't have information there. That's very important. Great. So we have data, but we cannot characterize what is going on here. There is a lot of molecular biology work trying to characterize this aggregation process. Is crazy. But what we decided to do was to come up with a model. We hypothesized what could be happening here and try to develop a very, very simple model. So the model that I'm going to show you, in principle, is going to be an stochastic individual-based model where every cell is going to have different states. Initially, all cells are going to be very, very hungry. They are starving cells, and they are signaling each other. So here, we have cells that are in the same identical state. They are all identical for us except for their spatial location in space. Just because they are sensing the signals of the other cells, they will, with some probability, transition to a state that we call an aggregating state. And it is very important that this transition from starving to aggregating is density dependent. It depends on how much signal you have around. These cells start to move, and as they start to move, the signal concentration starts to decrease. And some cells that didn't join the aggregate because this is a stochastic process, there is a probability, now they don't have signal and they are stuck. So imagine that you want to go to some bar, but you don't like going alone, and then everyone left, and then you stay in the hotel because now you're not going to go alone. So that is what we hypothesize. Of course, you can come up with different hypotheses that can also maybe explain this data, and if you have different ideas, we can talk about that, but that's what we proposed. What is very interesting is that when we, this is a stochastic process, so you can run simulations of these dynamics. You can also do a deterministic approximation for the mean values of the populations. That's what we did, and then write 3 differential equations. The interesting thing is that the differential equations are very, very simple. You can do analytical work a little bit, try to calculate some things. I can give you more details if you want, or you can find them in the paper. The interesting thing is that when you simulate these dynamics, the behavior that the model gives us is qualitatively very, very similar to what we see in the experiments. Of course, depending on how you parameterize the model, you will get different values of this plateau, different values for the number of cells that don't aggregate. But the existence of the plateau is, the model shows this plateau for a broad range of parameters, and you can even calculate analytically which conditions you need to have this plateau. So when you do that, you see that what really drives the existence of this plateau is a difference in time scales. So basically, if cells move very, very fast, they move fast enough to leave behind other cells, then you will have this plateau because they cannot keep up with the pace at which other cells are leaving. But if these cells move very, very, very slowly, they will eventually drag everyone in the population and make a perfect aggregation process. What is interesting is how environmental conditions are going to select for different aggregation strategies, act on different molecular, on different biochemical pathways within the cell and lead to different aggregation strategies. So this, this was supposed to be a very, very brief part, but I like this project so much that I always speak very slowly. So I'm going to go faster now in this part. So this is about microbes. We did many, many other things that I'm not going to show you, but we can talk about them. We mixed different types of these cells, quantified how the aggregation process was interfering, how the signaling of the different strains was interfering with each other to change the aggregation process. We found many interesting nonlinearities in this cross-signaling and many other interesting things that I will skip just for to keep things shorter. But something that I really like about studying biological systems, as someone coming from statistical mechanics, is to try to find similarities between systems. So how much can I apply to one system things that I learned studying a different one? And then once we were super happy because we had finished this project, took like forever, I was talking to, we were talking to some other students in the department, showing them our pretty plots, and they told us that they had some data on a completely different system that maybe could be interesting to check with this approach. So that's why, after talking to you about amoeba, I'm now showing you a video of the great migration of wild beasts in Africa. So this is just an example of a migratory animal. Sometimes, animals that live in seasonal environments. Usually what they do is that when it gets very, very bad in one place, they move to a different one. That's what's called a migratory strategy. Animals also includes humans. And then there is a lot of interest on trying to understand, first, what is that triggers this migration, especially when it is a collective behavior like this. How is this that these animals decide that all together they will go to some other place, because by going together they minimize a lot of risks regarding being killed by other predators, for example, or many, many other things. But another interesting question that relates more with the things that we are discussing today comes when you look at similar data that is similar to the one that I was showing you for amoeba. So the data that you're going to see in the screen now is data from 30 years. And every year, people counted how many animals you had in a given place. So imagine that these animals do like me. In winter, when in Germany gets cold and dark, they come to Brazil. And when Brazil gets humid and Germany gets sunny, then they go to Germany. So they track me, and imagine that there is many people like me, and they counted how many people like me stay in Germany in winter. So how many, if I have 1,000 animals, how many stay and how many leave, basically? So this is the number of animals that they decided to leave for an elk population in Canada as a function of the number of animals that they counted. Notice again the difference in the scales. So this is clearly, there is a linear correlation, but the slope is not 1, which means that not all animals leave. There is some animals that stay. They decide to stay in the worst place. Yes, so this is color code because this year there was a disease in the environment that I think changed a little bit the dynamics. So when you see it clearly in the slope. I think it was some extinction of some disease, some pest or something like that. And same here, you see this linear correlation. The slope is not 1. Again, 2 points that are colored differently. Because that year the season was particular, there was no seasons basically. The seasons were particularly weird, and it will come clear in a little bit for this case. It will become clear in a little bit for this case. So then the question is, if I have an environment like this, and now you will understand why I said that the slide mode mushroom was one of my biggest artistic creations. So imagine that this is the good environment. I have grass, and these my ungulates, they eat grass. So they are here very happy. And eventually the environment switches, and this is what happens, that some stay in the worst place, and the other ones decide to move. What is interesting is that the worst place, maybe it's very bad to host 1 million animals, but why not 200? And if I can avoid crossing the whole Africa, maybe I prefer to stay. very similar to what could be happening with amoeba before. So if we try to implement, again, this decision-making model, sorry, I was going too fast, trying to go fast. The idea, the hypothesis that ecologists, behavioral ecologists came up with to explain this pattern is precisely that at the end, the environments have different quality, but they may be a distribution of animals between the two patches that maximizes the food that everyone gets. Because if everyone goes here, maybe there is no food for everyone. And if they split a little bit, they manage to get some equilibrium. That is the hypothesis. But it's a hypothesis that is formulated at the population level without any detail on what is happening at the level of the individuals. When you try to explain what is happening at the level of the individuals, things get interesting and fun. Again, the easiest thing would be a coin flipping model, right? So basically assuming that animals decide to migrate with a given probability P, and if that were the case, we can calculate the distribution for the probability of having a fixed number of animals in one of the patches as a function of the total number of animals. It's one of the simplest probabilistic calculations that one can do. Great. So we have a model, we have a null model to test our data. So what we can do is we have the number of migrants, we have the total population size, we can calculate that probability for different animals, and this is what we get. And now we can do a further hypothesis. So this is the probability that one of these elks is going to migrate for all our different years. So you see that it fluctuates a lot, from 0.3 to sometimes 0.6, and there is a lot of variability in this probability, right? So there are different ways of thinking. How is that this probability is being chosen every year? That is the interesting question that we have now. One option is that somehow these animals have been living in this place for a very, very, very long time, and they have evolved this probability. It's a trait that has evolved to maximize the fitness of the animals in this seasonal environment. The problem is that if that was the true, if that was correct, there should be a probability that these, so if that was our hypothesis, then this probability should be more or less constant through years, right? Because if it's an evolutionary time scale, it doesn't make sense that I go from 1 to 0 if it's evolution that is fixing this trait. So one can do maximum likelihood estimation, which is a statistical technique to try to get a sense of whether one single probability can explain our data. And as you can see, what you can get from this lack of overlap between the red line and the blue line is that you cannot explain your full data set with one single value of this probability, which maybe was already obvious looking at the scatter data, but it's good to do things properly. You can do the same for the wild beast, and it's even more clear that the probability, one single probability doesn't explain your whole data set. So again, we are in the same scenario as we were before, asking the same question. And the question is, how are these animals deciding whether to migrate or not? So now you will see something that is very similar to what we had before, but now instead of having amoeba, and then I will ask you if any of you recognize what the cartoons for the amoeba were. We have the wild beasts that are in this seasonal range. They are trying to decide whether they migrate or not. Now they are undecided, but they are also hungry, so it's fine. We can call them hungry wild beasts. And at some point, some of them, they are sensing the environment. We're not going to say why. We're doing a very, very simple model. They are sensing the environment, and they are sensing what other animals do. And some of them probabilistically decide to migrate. And eventually, the social information and the information that they get makes them stay. They are not excited enough to leave, and they stay in that range. So again, we have one of these compartmental models. It's very similar to the SIR model if you want, but in this case, it's in terms of behaviors, where we have 3 different states with probabilistic transition rates. And what is really important is that this transition rate is dependent on the density of other animals. If you're interested in more details about how we are choosing this rate, probably to have this plateau, some of you are thinking that we need some step-like function. It is true. What we are using is a model that is very common in division of labor literature, which is called the threshold model, that basically says that when the information is above or below a threshold, individuals do different things, basically. Mathematically, it's a combination of theta Heaviside's function where you have a heaviside threshold for the environmental information and a heaviside threshold for the social information. Environmental information is how much grass do I have and how many people are eating my grass. Social information is what my colleagues are doing. So I'm not going to open the details of those modeling choices. What I'm going to show you is, again, I told you that that was a stochastic model. You can write the master equation for the number of animals in each state, and then you can calculate the mean value of each of the population sizes. I think Carmen explained you a little bit how to do that. At the end, you have a system of ODEs. Please remember that this is not linear because capital lambda is a function of U and M. So it's a piecewise linear system, which is nice to do analytical things. Anyway, when you integrate these ODEs and do your stochastic simulations, this is a very, very simple technical problem. But what is interesting is that when you look at the outcome of the dynamics, this partition between living and not living now is an intrinsic property of the aggregation process. And this is the important biological statement in the sense that when you flip a coin once, for example, and then you decide what to do or not. But when you have this kind of density-dependent decision-making process, you are constantly in time evaluating your environment and deciding what to do based on the information that you are collecting. Right? So it is not that the animals that don't live fail to live. It is that they are an intrinsic part of the collective behavior and they stay behind as an outcome of the collective behavior itself. It's a different way of interpreting things. And I think my laptop also wants us to eat. Yeah, okay. Anyway, integrating this model, we can calculate, again, we recover this plateau in the density of animals that decide not to follow the migration. Of course, we can tune and make hypotheses on which kind of information these animals are using. If they are only looking at the environment, they will hit what ecologists like the current capacity of the system. So if the system hosts 100 animals, 100 will live. And I'm not going to open up all the details, but basically what the take-home message that I want you to get from this plot is that you can analytically calculate the values of these plateaus and then relate it to the current capacity. Remember that this is the number of animals that the system can host, and it's the equilibrium that animals reach if they only look at the environment, of course. But when they also incorporate social information, which is what happens, it's what represents these other three lines, the purple ones and the red one, they can mismatch with the current capacity. That is the important message here. They can even leave more animals behind than what the current capacity of the system can host, and they can even leave less animals. So even you can, using social information, you can achieve a completely full migration. Why is this important and why is this interesting? Because there is a lot of manipulative experiments in these systems. Even at this scale, people manipulate the migration of ungulates. And they show how they can achieve equilibria that are either above the current capacity or below the current capacity, depending on how interfere with the amount of social information that animals use, or depending on how much grass there is in the environment. They can somehow make the environment better than what it would be naturally because of seasonality, and check how the animals decide differently. Just to finish, we have our model prediction for the number of animals that don't migrate. The complementary of that is the number of animals that migrate. That has a very, very strong linear correlation just because the number of animals that don't migrate is very small. So at the end, you're removing an epsilon from the total population size. And when you look at this partitioning process on data, it's also very nicely, it resembles very nicely to the same kind of qualitative behaviors that we see from our model. The residents plateau, and there is a significant linear correlation for the migrants. You can do the same for wild beast, and that's what I meant. Here, the season was so extreme that it's an outlier in terms of our statistical analysis. It's also interesting in terms of what we are proposing, because basically tells us that when seasonality changes and there is an extreme event, the animals adapt to it. So it's clearly something that something a little bit more plastic that they can adapt. And with that, I just want to finish with a bit, sorry, the references should have appeared later, but that way you can copy them, you have more time to copy them. So a little bit, a few take-home messages. So the first one is that, and I think this is what I wanted you to, I want to stay a little bit in your mind, is that imperfectly coordinated collective behaviors are frequent in nature. There are examples in locusts, in plants that synchronize their flowering time, and this flowering time is not perfectly synchronized. We can talk about systems like this if you want. The other important message is that these individuals that are not synchronized, and I should say coordinated maybe here so no one kills me, are part of the collective behavior. They could be part of a trait. So this is biological jargon, but it means that it can be something meaningful for the organism. That is really, really important. It's not just noise. And finally, that this could have some important ecological implications, which is the ultimate question that we are interested in. So with that, I just want to show you the people that we worked together on this. So Corina was my postdoc supervisor, and she was involved in both projects. And these are the dicty people, and these are the ungulate people. So thanks a lot for keeping your hunger under control, and yeah, I'm happy to take any question.
Okay, thank you, Ricardo. So before we migrate to the restaurant,
yes,
there might be questions.
Thank you, Ricardo, for this very, very interesting presentation. I wonder about for the elk population, ungulate populations, if those that decide to stay away from the group, are they the youngest? Are they are they, I know, ill, or is there any characteristic of the animals that decide that are away from the group?
Yeah, that's a very, very good question. And it's the question that I removed the 3 slides on that. So that's what makes this question interesting about partial migration, because so people have tried to characterize that a lot. Are they younger? Are they older? Are they bigger? Because if you are bigger, you're less likely to be eaten by a lion, so you just decide to go. The problem is that you don't find persistent patterns across a species. So for example, a very, very good example is for this wildebeest. You have different species of wildebeest, and the smallest ones, the ones that have a smaller body size, are the ones that migrate less. But when you look at ungulates broadly, larger body-sized ungulates are more likely to be migrants. Then depends also, there is many hypotheses. For example, older people, some people think that older individuals are less likely to migrate because they have fewer reproductive chances, so they just, that's one of the open hypotheses. I mean, even genetic, people have looked at whether these non-migrants just lack the genetic circuits that allow them to migrate. Let's say the state-of-the-art hypothesis is that it's a behavioral trait. Of course, it's influenced by body size, by many, many things. But underlying all these differences, the state-of-the-art hypothesis is that it's something behavioral as well.
More question.
Thank you for the talk.
Thank you.
It was 2 different cases, but very interesting because, at least for me, I'm a biologist, I see that the correlation is much more on hunger of those 2 systems. But I was going to ask if you have ever seen other types of migration or collective motion behavior on biological systems that are not driven by hunger, for an instance, for different behaviors, for an instance, migration for reproductive matters.
Yeah, so I actually should say that in the case of ungulates, for example, it's true that the environment gets poor, so they cannot feed, but and then they migrate to reproduce, and then they stay in the other seasonal range, and then they come back with their calves already grown. There is a very, very, to me, it's a very interesting example of collective decision-making migration, and it's, I can send you the paper, it's a PNAS paper. and it's from people from Blanes, in Girona, in Spain. And the system that they were analyzing is a population of birds, a colony of birds. And they have data on many, many years. And the perturbation that the system had, at the end, hunger is like, I mean, consuming all the food is a perturbation, no, in your system, is the arrival of a cat. So there is a cat that gets to the colony and starts killing like small birds, and they track the number of animals, the number of birds in the colony, and they see that eventually, after the cat has been around for a while, there is a non-linear response in the system that they all decide to do, and they quantify, if I remember right, they quantify even like the non-linearities in the decision making. And so in general, every perturbation that affects your system and then you need some quorum to leave the place, it's
really nice talk.
Thank you.
My question is regarding the mechanism behind these heaviside-like behavior. Is there like a molecular signaling mechanism that has this behavior? And is this the results of the model sensitive to the change of the curve? I mean, if you get something less restrictive as a sigmoid or something, does it behave the same way?
Yeah, so those are very good questions. Starting with the easy to answer one, the easier to answer one. So if you choose a sigmoid function that is stepped enough, you keep the same behavior. The main difference is that you cannot, for example, so in this figure here, which is one of my favorite results of this project, the blue line is an analytical solution in the limit of an infinitely large population, where you can really prove mathematically that this plateau exists and calculate its value. And you can do that just because your system is piecewise linear. So to do that, you need a heaviside function, so it's mathematical convenience. Regarding the molecular pathways, indeed there is a lot of work to characterize those. The big problem is that for many, many years, people thought that these cells are noise. And that was a big experimental challenge that we had. Even experimental protocols, they are designed to eliminate those cells. You know, so for example, I mean, I didn't show the data, but when you do this experiment, changing the properties of the soil where you are doing your experiments, the agar concentration, the gelatin, this changes a lot. Of course, because the signals diffuse less, the mechanical properties, the stiffness is different. So people were, for many, many years, were doing experimental protocols so they could get rid of this noise. So they were not looking at the molecular mechanisms that drive this decision making. And that was, I mean, until we got this published, in every single journal, they were asking us that without molecular things, reject. So I think the interesting now biological question is, that's why I was super careful saying this is a hypothesis.
Okay, so thank you, Ricardo.
Thank you.
For the very nice talk. So now we have a lunch break, and we'll be back at 2:30.